Hello and welcome back to the Omics Logic Data Science program. In this session, we will review classification and metrics. We will discuss feature selection, revisit some of the challenges and use cases for feature selection. We'll review the content and assignments moving forward and discuss the generalized regression models, which are the main topic of today. First, let's see what we have covered so far. In the previous sessions and assignments, we learned how to load, transform, and visualize data. We talked about statistical evaluation of a dataset and analysis for hypothesis testing. We also spoke about performing exploratory analysis of a dataset and understanding its structure. We talked about the link between molecular features, sample groups, and how to identify important trends in data, and had a discussion about various clustering methods and ways to evaluate their use for a given dataset. Finally, we talked about classification and feature selection. Briefly, let's review some of these topics to understand how they are all connected to each other and what were the major outcomes from each one of these exercises. First, let's talk about data mining, dimensionality reduction or embedding, and clustering. We briefly covered three different methods: PCA, which reduces dimensionality of the full dataset to preserve data variance. It uses Euclidean distance between samples based on all features and is used to find such projections that retain most distances found in the original space. Evaluating distances in the projection have to include the explained variance of each component. That way, if PC1 is 40% and PC2 is 10%, distance on the x-axis is 4 times greater than the distance on the y-axis. The second method we talked about was MDS and NMDS, multidimensional scaling and non-metric multidimensional scaling. So sometimes we might be interested in a dataset that we suspect has a composition with abstract measurements of diversity and change between distinct groups of samples. The goal of an MDS is to collapse information from multiple dimensions into just a few so that they can be visualized and interpreted. Unlike many other techniques that rely on Euclidean distances, NMDS uses rank orders, which show closer points related by their rank in the respective group. Reading an MDS plots is simple. Objects that are ordinated closer to one another are likely to be more similar than those further apart. But the scale of the axes is arbitrary, as is the orientation of the plot. One measure of NMDS quality is a measurement called stress. Higher stress, usually about 0.2, should be interpreted with caution, and stress above 0.3 is highly suspect. The next method we talked about is t-SNE, or t-distributed stochastic neighbor embedding, which is a technique for dimensional reduction that is especially good for high-dimensional datasets with non-normal distribution. For example, we used it for single-cell data that is zero-inflated. Specifically, it models each high-dimensional object by a 2- or 3-dimensional point in such a way that similar objects are modeled by nearby points and dissimilar objects are modeled by distant points with high probability. The distances refer to perplexity, which is a measure for information that is defined as 2 to the power of the Shannon entropy. Perplexity may be viewed as a knob that sets the number of effective nearest neighbors. Loosely speaking, one could say that a larger or a denser dataset requires a larger perplexity. Typical values for the perplexity range between 5 and 50. One of the challenges in such scaling is that we don't intuitively understand these distances, and so we have to rely on some rules of thumb when we know the number of samples, the numbers of features, and what our approximately data looks like. One way that you can dig deeper into these methods is to apply them to various datasets. So we provided you with 3 different types of data. Gene expression data for distinct types associated with tumors in different tissues, mutation frequency data from viral genomes sampled from various geographical locations across time, and a microbiome dataset with operational taxonomic unit abundances across several types of conditions. Using your understanding of the methods that we discussed, like PCA, NMDS, t-SNE, and UMAP, you can see how dimensionality reduction can make data interpretable, revealing patterns that you can build upon in your final comparative analysis. Importantly, one takeaway was for you to see the relationship between the number of features, the number of objects, the amount of noise that the data has, and the specific types of labels that we can use to understand the explanatory value of such a dimensionality reduction. What is especially interesting to see after we understand how dimensionality reduction algorithms help us find patterns in data and visually interpret them is to further explore clustering as a way to organize these patterns into distinct groups or clusters. With so many algorithms for clustering that exist, developing a sense of how these algorithms perform with various datasets can help you select the right methods for the task at hand and know how to evaluate the results. Clustering can be used in several different ways, helping us find meaningful patterns that explain phenotypes or even uncover hidden patterns that can lead to biased interpretation. What's important is to always test our assumptions, working with the data and trying to understand what might lead to observed results that we get from the analysis. Scikit-learn has a collection of such methods implemented in a standardized and easy-to-use framework with parameters and outputs that we can use to understand how each one works and what are the specific ways we can test and measure the success of our analytical approach. This includes methods to explore data structures, find optimal number of clusters, explore inter- and intra-cluster variation, and evaluate how well clusters match our expectations if we have labeled data. Importantly, we can test the results visually using dimensionality reduction methods with 2 or 3 projections to help us spot unexpected variation. We can also use clustering to study the features or the objects that we have in our data. For example, when we use clustering to study features in our data, we have to transpose the data frame. In our example, we can explore the gene expression profiles to find closely related patterns that differentiate between genes responsible for various biological functions. In this data frame, our rows are genes and columns are samples. We can transpose the data frame and look at it in a different way. We can look at samples being the rows and genes being the columns. However, since the number of genes we have is much greater than the number of samples, linear dimensionality reduction gives us a somewhat meaningless picture that explains a lot of data variance without any distinct patterns, which means the patterns might be more complex than expected with many smaller groups of genes that are linked together. In such cases, nonlinear methods like t-SNE or UMAP can be useful. Non-metric embedding methods like t-SNE and UMAP can be used for high dimensionality data together with density clustering like dbScan, HDBScan, optics, and spectral clustering to identify closely related clusters of genes that can have a biological interpretation. Here you see a visual of dbScan after dimensionality reduction with t-SNE and UMAP. Importantly, UMAP can provide us with multiple projections, while t-SNE is limited to just a few. Knowing how to use different methods and what are the limitations of some of the clustering methods, we can arrive at an appropriate combination of dimensionality reduction and clustering to identify meaningful groups. In the exercises, you should be able to explore noisy and biologically meaningful clusters of genes that would lead to additional questions about the datasets as you mine them. Evaluating identified clusters in the data can be biologically interpretable, like this cluster with high intercluster correlation coefficient that all maps directly to a single biological function of ribosomal genes. We can again spot variation that can influence clustering and reveal important abnormalities, like an outlier sample or group of genes. We can also develop metrics to compare clusters to each other to understand how gene sets are related. In conclusion, data offers a lot of insights as we play with it and mine it for patterns. These patterns can be revealing about the complex relationship between objects and features in our data. Insights can be technical or interpretable in a biological way. Most importantly, these patterns are important to study and understand using appropriate methods, settings, and evaluation metrics that we have learned about so far. Now let's also review a different set of methods that we covered related to the task of classification. One way to think about our data is to view it as a combination of noise and signal, which could be explained by a model. A model in statistics can be a function and a set of coefficients that follow certain assumptions. For example, we have a general understanding of probability, which we can apply to hypothesis testing, or we can learn to extrapolate what we see in data with some measure of confidence. In machine learning, algorithms and statistical principles can be extended further. A model in machine learning is the output of a machine learning algorithm run on a specific dataset. A model represents what was learned by a machine learning algorithm. The model is the actual thing that is saved after running a machine learning algorithm on training data, and it represents all of the different rules, numbers, thresholds, and any other algorithm-specific data structures required to make predictions. Once the model is trained, it can become a method that we rely on to deal with new data, or it can be studied to really understand what's going on inside the dataset and why we find certain patterns. To train the model, we have to label the data by using phenotypic information like cancer stage, subtype, or risk factor. An algorithm learns this target mapping function from training data. The learning can be summarized as a learning of a function f that maps input variables x to output variables y. Assumptions about the data and possible function that models relationships can greatly simplify the learning process. A learning model that summarizes data with a set of parameters of fixed size, independent of the number of training examples, is called a parametric model. No matter how much data you throw at a parametric model, it won't change its mind about how many parameters it needs. Non-parametric methods are good when you have a lot of data and no prior knowledge, and when you don't want to worry too much about choosing just the right features. In many cases, advances in computational technologies made many non-parametric methods that were too costly to train accessible to anyone on sizable datasets. Decisions about assigning a sample to one class or the other follow a parametric or a non-parametric approach. SVM and random forests are both non-parametric approaches that rely on data quantity. The output of a model can be evaluated for accuracy, precision recall, and other representations that we went over in exercises. Importantly, we saw how for most non-parametric methods, training and test data that accurately represent the conditions we have plays a major role in the quality of a model overall. Since many datasets have some overrepresentation for various classes, it is important to understand these metrics in the context of each class. For example, if the total number of samples you have is 10, and one class is only 2 samples, then an 80% accuracy still is pretty bad for that one class. We can also see how various non-parametric models are affected by the number of features. One way we can do this is by running a dimensionality reduction method that can retain multiple projections that are used for training. As we can see here, UMAP already does a great job at retaining the expected data structure by maximizing distance between subtypes of samples in our dataset. In one of the exercises we reviewed, the number of UMAP projections has an impact on the accuracy of classification for various models. Support vector machines performs best overall. That is because we never reach a point where the number of projections is greater than 30. Random forest achieves perfect accuracy at 5 dimensions, decreasing with more number of dimensions that are being added. KNN seems to perform better with higher number of dimensions, but does not achieve good accuracy overall. What are some of the conclusions that we should have about these classification models? Non-parametric methods are affected by data used in training and test datasets. Noise to signal ratio influences classification quality for models in different ways. The number of features and what these features represent, whether they represent noise or actual signal, like in UMAP or PCA projections, is also critical for the quality of the model. Reducing the number of dimensions helps some classification models. Dimensionality reduction helps us understand model quality intuitively and link it to a biologically meaningful interpretation. One of the useful ways to study a parametric model is to understand the relationship between specific features and class predictions of a model. In the exercises we reviewed, you would have seen many times how a dimensionality reduction algorithm like PCA is applied to thousands of genes or just a select few. When thousands of genes are present, many of them could be considered noise that actually prevents accurate separation between known groups. So we might only spot the most significant variance in data. In the case of the cell line dataset, we only see a separation into two major groups that are the most different from each other. Classification that looks for separation between defined classes will learn the difference between classes based on coefficients. We can take the top feature importance from models like SVM or random forest. Performing PCA on select features will reduce noise, thus increasing variance explained by top principal components. The reduced dataset of 15 genes has an overall variance explained of almost 80%, compared to only about 30% for the full dataset. These 15 genes were selected based on a model output which assigns accuracy and impurity measures to each gene. Such measures can help select the best features that can separate between classes and optimize classification. We can also view PCA loadings that show how each gene profile contributes to variance. Direction of each loading can be used to understand the way such feature contributes to separation between labeled groups in our data. The random forest classification relies on randomly selected portions of data and many classification trees. Majority voting helps select the optimal decisions for a classification. The data points that repeatedly show increased accuracy and decreased impurity between classes can be selected as the top features that contribute to the model accuracy overall. In scikit-learn, these are termed feature importance based on mean decrease in impurity and feature importance based on feature permutation. Support vectors are data points that are closer to the hyperplane and influence the position and orientation of the hyperplane. Using these support vectors, we maximize the margin of the classifier. Deleting the support vectors will change the position of the hyperplane. These are the main points that help us build our SVM. In linear SVM, the resulting separating plane is the same space as your input features. Therefore, its coefficients can be viewed as weights of the input's dimensions, and we can use these weights to select top features that are responsible for model accuracy. Now that we've completed this review, let us turn to another important topic for machine learning, parametric classification and regression. So let's now speak about generalized linear models. First, let's talk about linear regression. Variables are terms we use to define our data in an experiment. Any externally controlled variable related to data collection is called an independent variable. It is manipulated by what we intend to study. The data we collect are the dependent variables. These variables represent the measurements we take of the conditions in the experiment. Oftentimes, we would like to establish a relationship between changes in independent variable to establish the relationship of measured dependent variables with the cause we control. Sometimes, this cause can be indirect and therefore not a causal effect, even though it appears to be so. Nevertheless, establishing a non-random relationship between the two is important. Let's say we collected data on eye, color, sex, age, height, and weight of people. We hypothesize that some variables here are related and some are not. We also think there are exceptions to the rule. The age and height or weight are related, but maybe not always. Relationships observed in data can show which variables are related or unrelated, and if related, then by how much. For example, weight and height seem to be logically related. Are they in this data set? If we plot a trend line, we will find that the trend line goes up. The bigger the height, weight is also increasing. But the dots are not too close to the line. Some dots seem to be far away from the line. Is there a way to quantify this relationship we found? To study the relationship, we can use a coefficient of correlation, or R squared. This measure shows how well the data in our dataset is explained by the correlation line drawn through this data. In this case, we get an R squared coefficient of 41%. 40% is okay, but not perfect. Our conclusion is that height and weight are correlated, but not perfectly. Such relationships can be of several types: positive, negative, and nonlinear. Linear regression is a way to further model such a relationship to determine a mathematical function explaining the relationship between a dependent and independent variable in the dataset. If there is a relationship, such a function is predictive and will inform us how the independent variable will change if we change the dependent variable. The goal of regression analysis is to find the right coefficients of this formula that minimize predictive errors based on the data that we have. The regression formula has both the dependent and independent variables in a mathematical equation, plus a constant intercept and a coefficient. The dependent variable is related to the independent variable with additional considerations: a constant intercept, or where the line starts from the slope of the regression line, also known as the coefficient. To get these variables from data, we use the least squares approach. It helps measure the distance from each dot to a line, finding the line that best fits the data. Simple linear regression is an approach to model the relationship between a dependent variable and one independent variable. The linear regression model assumes a linear relationship between the input and output variables. If this relationship is present, we can estimate the coefficients required by the model to make predictions on new data. Same principles can be applied in ANOVA, where variables are factors, and factor regression analysis. Factor regression analysis is an example of multiple linear regression that uses a linear function to predict the value of a dependent variable containing the function for n independent variables. It can also be applied to multiple independent variables. The entire set of data points can be evaluated for fit to the regression function. To use linear regression, we add another parameter called E or epsilon, which is normally distributed and centered around the true regression function. This is a random component which represents the data fit, or distance, between the data points to the regression function by non-random or structural components. As a result, the fitted model can be used for prediction of continuous or categorical variables. For categorical variables, we can use the regression approach to model a relationship between probability of category for a given observation and the continuous data we have. If we imagine having just one type of variable, for example BMI, we can use the label of each observation, some clinical diagnosis for an individual, and data X, in our case BMI, to model a relationship between these two variables using a distribution of samples around two centers. Let's call them N for normal and D for diabetes. A Gaussian curve of probable distribution for the given set of data has to be modeled using an assumed distribution, its center on the axis, and the sigma, or how wide it is. In statistics, maximum likelihood estimation is a method of estimating the parameters of an assumed probability distribution given some observed data. We have to determine the distribution curve of probability for samples with each label, or find the sigma and center for each Gaussian curve. For linear regression, we can use the least squares methods to determine the most fitted linear regression function for the model parameters, or find the separation point where the relationship between probabilities is equal to 1. For example, if the group of people in a dataset with low BMI is less likely to have diabetes, and the group of people with high BMI is more likely to have diabetes, we can calculate probability for each group to have patients with diabetes and learn the relationship between BMI and diabetes in terms of probability. Using such a model, we can predict which class any new observation belongs to. Quality of our prediction will be determined by the fit of our model to data. That is where we need to be mindful of the assumptions in our parametric model. The probability curve is a sigmoid that can be modeled by a logistic function. The curve can be used to make a decision about any data point we might want to do prediction on. The same can be done for more than one variable, for example with x1 as BMI and x2 as blood glucose. Same as with one variable, we will now have to estimate a probability curve based on some distribution, where the Gaussian multivariate distribution is used. Using the same set of linear equations, we can now fit the model to our data. For any new point, we use the data coefficients to identify the most likely class it belongs to given data x1, or its BMI, and x2, or its blood glucose. This model for probability is called logistic regression, which is a statistical method that can also be termed machine learning in its algorithmic form. The goal is to model the probability of a random variable y being 1 or 0 given experimental data. Regression modeled using the logistic function can be transformed to linear regression through a logarithm on both sides of the equation, which is also called log transformation. How do we actually make a decision? For a test instance X, we say yes if the probability is more than 0.5 and no if it is otherwise. We call 0.5 the decision boundary. In scikit-learn, logistic regression is a part of the linear model library. That is because logistic regression uses a linear function where function coefficients have a linear relationship. To use the model, we need to specify whether we have a binary problem or a multi-class problem, where we can use one versus the rest scheme or the multinomial scheme, where multinomial refers to multi-class. We can also specify the solver for this model. For example, LBFGS stands for Limited Memory BFGS, the Broyden-Fletcher-Goldfarb-Shannon algorithm. The solver is an algorithm to find the optimal fit, in this case applied to gradient descent. Gradient descent is a numerical approximation of the ordinary least square solution. Ordinary least squares is the analytical solution for a linear regression problem which finds the least value of squares of the error. Applying this to linear regression is fairly easy because of found coefficients and linear equations. Gradient descent is an alternative to the OLS solution by iterative method. The basic idea of gradient descent is to find such a portion of the error curve using a step which can help us find the minimum value, assuming that the curve is nonlinear. For each step, where step size can be set by the user, we can approximate the curve as a straight line and calculate the descent in weight until we reach the global optimum for this approximation. Such a solution to maximize fit or minimize errors in fit for our data can be applied to generalized linear models and even nonlinear classifiers like support vector machines. Generalized linear models are called generalized because they assume a distribution as the basis of their link function. Most commonly used ones are the linear, logistic, and Poisson regression models. To summarize, the GLM consists of three components. The random component, which specifies the conditional distribution of the response variable y, given the values of the explanatory variables x. The probability distribution, which generates the observed variable y. The systematic component, which specifies the linear combination that explanatory variables x are fit into by estimating b in various ways. This is also called the linear predictor, a linear combination of parameter b and the explanatory variable x. Finally, the link function, which specifies the link between random and systematic components. Link function literally links the linear predictor and the parameter for probability distribution. Linearity means y is a linear function of parameter b. Regression is a powerful method for both classification and regression problems. It can be used for continuous variables or for categorical variables we would like to predict. Categorical prediction can yield weights for observed variables and independent observations. This is a typical case for ANOVA or factor regression analysis, where we organize our observations into groups based on some factor and find a probability for each variable in relationship to an effect of that factor. In classification, the categorical value is predicted based on the distribution of probabilities modeled using a model that was fit to our data. This model can learn from a given dataset and predict whether a new dataset can be classified. A model like that is called a classifier. For continuous variables, regression allows us to learn from data and predict the next variable by learning about data structures. This is often useful when we have missing data in a set of continuous variables or in time series data where previous events can tell us something about the future. This type of a model is called a regressor. That's all for today. In our next session, we'll be speaking about network analysis.